Hi, everyone. Welcome to our talk titled FlyBrainLab Interactive Computing in the Connectomic and Synaptomic Era. My name is Tingkai Liu. I'm currently a PhD candidate in Columbia University in New York City, and I'm presenting this work on behalf of my research advisor, Aurel Lazar, and my lab mates, Mehmet Turkan and Yiying Zhou. We're very excited to be given this opportunity to share with you some of the news about fly-brain neuroscience and how we have leveraged the JupyterLab platform to really accelerate the discovery of the functional logic of the fly brain. And to begin breaking down the title of our talk, I'd like to talk about fruit fly brain in the connectomic and the synaptomic era. And we will begin our discussion by talking about the fruit fly and the fruit fly brain. Fruit fly is known to many as a common household pest that has a natural affinity to decaying fruit, and therefore the name fruit fly. What many people do not know is that fruit fly has really been at the forefront of scientific discovery over the past 100 years, contributing to 5 different Nobel Prizes for discoveries ranging from genetics to immune system. We now know much about the behavior of the fly, or in some sense, the consequence of the function of the fruit fly brain. And these are, in some sense, analogous to the cognitive capabilities of humans. Already showing this picture are a few, namely the ability of flight and the ability to locate and navigate to food sources. In recent years, we have seen an explosion of data that has been generated on the structure of the fly brain. Shown here in the video is a sparse collection of some of the neurons in the fly brain that we work with in our research. And to give you a sense of the scale at which these data has been produced, we highlight 3 different data sets that we work with. From left to right, we have fly circuit data set, hemibrain data set, and larva data set, covering anywhere from 10 to 40% of the adult fly brain on the left side and the larva fly brain on the right side. These provide information about not only the structure of the neurons, but how neurons connect to each other on single synapse levels. And we expect the 100% of the fly brain to be mapped out within the next year or 2. And this together will provide the single most complete information about any animal brain that is capable of complex behavior. And these, to better illustrate how we use these data and what these structural information typically look like, I would like to give you a demonstration using one of the in-house web applications that we have developed called NeuroNLP. And here's a screenshot of NeuroNLP. This web application has 3 components. On the left side, we have the info panel. On the right side, we have the 3D visualization window. And on the bottom, we have a natural language processing query bar that is currently showing show L1 home. And L1 home is a neuron in the adult visual system that we are visualizing first. Typing in this query sends it to our server-side backend, which is processed from plain English query to database query. And the result of this database query is returned to the user in an example interface like this kind. And not only is the structure of the neuron returned to the user, as visualized in 3 dimension in Scion shown on the right, the database also returns metadata and connectivity information about the neuron. And now we can use the connectivity information to construct a neural circuit that is downstream of this neuron, L1 home. And we start by adding the most significant postsynaptic partner, MI1 home, that has 150 synaptic connections to L1 home. And we can continue doing this and adding more and more neurons. And eventually we can end up with a subcircuit like this, with 5 neurons, L1, MI1, and 3 downstream neurons of MI1 that are T4 neurons. And together, this forms a view of the kind of structural information that we work with: neuron structures, their metadata, and also their connectivity patterns. So in summary, on one hand, on the left, we have the structural information that we just described. And on the right, we have the accumulated knowledge about the function of the fly brain. And on a high level, about the behavior repertoire of the fly brain, its ability for learning and memory, for feeding, etc. And recently, in the past few decades, we also have on single cell level how some of these neurons actually respond to stimulus under specific conditions. And what our job is as computational neuroscientists is really to connect structure to function through models. And these computational models need to be topologically grounded and constrained by the actual underlying neural circuit in the structural side, but it's also required to reproduce some of the critical characteristics of the function of the fly brain. Now, in the past, our lab has produced a collection of computational tools that target all three building blocks of this structure to function paradigm. We have described the NeuroNLP for the structure side. We also have another web application called NeuroGFX for the functional side. And we have a Python simulation engine called NeuroKernel that allows you to simulate very large-scale neural networks for the model side. However, these tools have been developed independently and in some sense separate from each other. And data interoperability between nodes, between these tools, have traditionally been fairly difficult. And I should note that this is not just the challenge that our lab faces. This is a very common issue in the field of neuroscience where different data provided at different time using different technologies by different groups are often in a different format with accompanying tools that are specifically made for this data type. Around 2018, to address this issue, we started thinking about integrating all the tools that we have developed into a unified framework. And coincidentally, this is also the year that JupyterLab came out with its beta release. And we immediately realized that this really is the perfect platform for us to do this integration across data modality. And starting JupyterLab 0.31, we have leveraged the extension ecosystem of JupyterLab to develop a unified integrated platform that combines all data types into one giant JupyterLab extension. And thus, the FlyBrainLab interactive computing platform was born. The main access point of the FlyBrainLab platform, and what I would like to highlight today in this talk, is what we have built on top of the JupyterLab front end that is called NeuroMynerva. Here's a screenshot of a typical interface of NeuroMynerva that we use in our day-to-day research. On the screen, you see a couple of widgets. For structural data, we have two widgets that replaces the NeuroNLP web application that we have just mentioned. These widgets show three-dimensional morphology of the neurons in the middle and the info panel showing the metadata and the connectivity data as we have seen before. For functional data, we have a dedicated widget that replaces the NeuroGFX widget. And for computation, we simply rely on the native JupyterLab extensions with notebooks and local file access to manage our simulation code as well as data. Together, the NeuroMynerva interface that is built upon the JupyterLab extension system provides a single integrated solution that solves a bunch of design requirements that are specific to scientific research in neuroscience and not just in fly brain research. The first requirement that comes with neuroscience research is independent rendering of data types, as we had in our individual web applications previously. Here, this is represented as two widgets on the right side, NeuroNLP for structural data, as we have seen before, and NeuroGFX, which is the web application that I was mentioning that we have developed to represent functional data. In order for these widgets to obtain the data, they need to interact with some kind of server-side backend that hosts our key assets, like our database and our natural language engine that converts plain English queries to database queries. To support access to our backend services, we chose Crossbar, which is an application that implements web application messaging protocol. And this allows users to access these server-side assets over the internet. And to improve the accessibility of our platform, we would also like to support data coming not just from the server-side backend, but also from other sources, like, for example, local files. Moving to the next design requirement, we would like to have multiple instances of the same widget rendering different data type in order for us to compare data between, for example, the adult and the larva. We would also like to have, of course, data interoperability between the widgets. And this would give us the ability to, for example, not just look at the activity pattern of a given neuron, but also the local connectivity that gives rise to such activity pattern. And lastly, we also need programmatic access to the different data types that are being rendered in the widgets. Given these constraints, here's an illustration of the extension package that we came up with. By default, every widget in the browser has an associated kernel that is connected to the backend server as an independent crossbar client. The bidirectional communication from the widget frontend and the kernel in the Jupyter server is done through the kernel's request execute function and the JupyterCon protocol. Importantly, we designed this architecture really with data interoperability as a top priority. And the user can now do data integration by simply changing the kernel associated with one widget to another. And using multi-threading on the Python kernel, we can support uninterrupted parallel backend query using the same kernel. In creating this package, we realized that there were a couple of challenges that we have to overcome. And the first challenge was that we want to support both the browser-side entry point and the kernel-side entry points. In iPad widgets for Jupyter Notebooks, traditionally we have kernel-side entry points that allows users to create these front-end views by instantiating iPad widget instances in the Python kernel. Now, in JupyterLab ecosystem, we typically have browser-side interactions like command palettes as entry points. In situations where data can be either obtained from server-side queries or in the kernel space or through browser interactions like reading a local file, we really need to support both types of interactions. Following the example code on JupyterLab, we designed our features of the widgets around the commands exposed to the overall Jupyter application on the browser side. To allow users to spawn these widgets like they would in IPyWidgets, we used a package called FlyBrainLab that is developed by Jeremy. And however, IPyWidgets are only functional in notebooks, and it would be great to have this type of support from anywhere that has a kernel connection. Another major challenge when it comes to developing a large collection of widgets for JupyterLab is that the build process for JupyterLab can be a little bit long. Since all the extensions that you are working on need to be built together with the entire JupyterLab application. One of the major advancements that's coming with JupyterLab 3.0 is module federation with the latest Webpack, and this is going to solve this problem once and for all, and life is going to be great. What we have settled on so far in the interim is to have a single extension that's built into the JupyterLab application. And this extension injects require.js into the browser at runtime, and it also loads a separately built widget package through HTTP request. And what this will allow us to do when we're developing these widgets is to just build the widget independently somewhere else and refresh the browser, and the latest built widget will be used in the new JupyterLab instance. The last challenge is that we wanted to support easy data synchronization between the browser side and the kernel side. And traditionally in Jupyter Notebooks, this is done very automatically with the JupyterCon's protocol. And what we have ended up doing in the JupyterLab environment is to do this manual synchronization through callbacks and using JupyterCons. And it would be great if there's a standardized paradigm that borrows the design philosophy from Jupyter Notebook into the JupyterLab extensions. Nevertheless, we overcame these challenges, and what we ended up with this NeuroMynerva and this FlyBrainLab platform is really an integrated computational platform that allows users to analyze, evaluate, and compare FlyBrain circuit models. And importantly, the ability to spawn multiple widgets or even workspaces in JupyterLab enables easy comparison between different brain circuit models. And in this talk, I'm going to go over one particular example where we compared different models of a particular brain region that's called the central complex. Now, before I go into the details of the central complex scientific aspects, I would like to give you a video demo of how a common work session would look like when we were comparing these models side by side. So what I'm showing you here is 3 columns corresponding to 3 different NeuroMynerva or FlyBrainLab workspaces. Each column has 3 windows. From top to bottom, there's a Jupyter notebook, there is a NeuroNLP 3D visualization widget, and there is a NeuroGFX visualization widget that shows you the executable circuit. And the Jupyter notebook allows users to query the data from the server-side backend and also perform simulation and manipulation of the circuit, etc. But this integration between widgets, what this allows users to do is interactively go into these executable circuit, change, for example, parameters of the neuron models inside the circuit, or add or remove neurons or synapses in the circuit interactively. And these changes are reflected in the Python kernel directly by this bidirectional data binding. And after you are satisfied with the circuit manipulation, you can now compile the circuit within the notebook using our Python API, run the circuit, and then visualize the output directly within the notebook and compare them side by side. Now, I would like to put this video demonstration into more scientific context. And to do that, I would like to describe to you what central complex is, and particularly the key component of the central complex neural circuit that we are highlighting. So all the models that you have seen on the previous slide are really describing all the neurons that are shown on the left side of this screen. This central complex brain region is in the center, as the name suggests, of the fly brain. And it functions as a compass of some sort for the fly brain, supporting important functions like navigation and localization of the fly with respect to its surrounding. And importantly, and very interestingly, there is a substructure of the central complex called the ellipsoid body that is shown here that has the shape of a torus. So not only does it function like a compass, it also looks like a compass. The ellipsoid body, as you can see here, is comprised of a bunch of different neurons forming a compartmentalized partitioning of the torus, with a single bump activity pattern commonly seen as shown on the bottom right. This single bump activity pattern has been shown in 2015 by researchers from the Genelia Research Campus to be able to track the location of a moving visual stimulus, essentially providing a neural representation of the moving object with respect to the position of the fly. In the setup in 2015, the researchers placed the fly inside a cylindrical virtual reality arena, where a visual pattern shown here as a single vertical green bar is presented on the perimeter of this cylinder moving counterclockwise or clockwise. Looking top down, the input pattern is also forming a torus that in this case would move clockwise 360 degrees first and then move back 360 degrees counterclockwise to the original position. The researchers used calcium imaging technology and measured the activity pattern of the ellipsoid body as the visual stimulus is moving. And if you plot the angle of the input pattern and the angle of the highest activity pattern of the ellipsoid body, you will get a waterfall plot as shown on the left, where the left figure shows the position of the input bar and the right figure shows the activity pattern of the ellipsoid body. And as the input stimulus moves clockwise, you see that activity pattern of the ellipsoid body also tracks the position of the visual input. And as the stimulus goes back 360 degree counterclockwise, we have this zigzag pattern. Now, this striking correlation between the localized activity pattern in the ellipsoid body and the visual input has intrigued many researchers. And the reason is not just because of this striking correlation, but really also because that not only do we have this functional recording data, we also have very detailed connectomics information about how the neural circuit looks like. And because of this combination of the structure and functional data, central complex became fertile ground for modeling work that seek to explain its ability to track the visual input. And here, we use FlyBrainLab to implement, simulate, and compare 3 of these models to look for the common denominator between these models, which hypothetically will be the fundamental building block of this tracking behavior of the ellipsoid body. We begin by looking into the model description of the ellipsoid body in the literature. The first one shown here is a work that was published in 2017 by a former member of our lab. From top to bottom on the left column, we have the neurons that are described by the model, as well as a detailed circuit diagram on the bottom. Using a slightly different input stimulus, now just with one additional stationary bar that is lower intensity at a fixed angle, 60 degrees to the fly's head, we can simulate the response of the ellipsoid body. And since the ellipsoid body activity pattern is expected to track the location of the input pattern, the response pattern still reflects the input pattern in this zigzag format. And we can repeat the same process of model development and simulation for 2 other models of the central complex, shown here in the middle and in the right columns. These 3 concurrently published models each describe a different subset of the neurons in the central complex, as you can see in the morphology and in the circuit diagram. And finally, using the same input to all 3 models, we perform a comparative study of their input-output behaviors. We see that although there are some slight differences in their response patterns, all 3 models are able to track the behavior of the ellipsoid body fairly well. We then look at the shared circuit features that these models are describing, and that allowed us to pinpoint a global inhibitory neuron that is really the key circuit feature that it gave rise to this tracking behavior of the ellipsoid body. And we encourage you to read our preprint for the details of our findings. And I would like to just conclude by emphasizing that in this era of unprecedented data availability in neuroscience, we believe that the opportunity is really ripe for us to go from structure to function of the fruit-fly brain. You can read more about our research by following us on our Twitter, following us on our GitHub, or checking out our website. And thank you very much for your attention.